In this video, we'll look at both ovule and pollen development. We'll start by moving inside the ovary to examine ovule development. You'll be observing a prepared cross-section of a Lilium ovary under the microscope. This is where the female gametophyte develops, deeply protected inside the sporophyte tissue. As you observe, I want you to think about how angiosperms have reduced and protected the female gametophyte compared to earlier plant groups. Inside each ovule is the megasporangium, also called the nucellus. Within the sporangium is a microspore. That cell undergoes meiosis, producing 4 haploid cells. 3 degenerate. 1 survives, the megaspore. The megaspore then undergoes mitosis to form the reduced female gametophyte. Before observing, think about this. Why reduce the female gametophyte? What advantages might come from minimizing tissue and keeping it enclosed within the ovary? Consider efficiency. Consider protection. Consider speed of development. Write your prediction before continuing. We're going to observe the ovary cross-section at both scanning and higher power. On the left image, locate the ovary wall and identify the locule, the internal chamber. Inside the locule, you should see multiple ovules attached along one side. In higher power, in the right image, one ovule is centered. Identify the megasporangium, also called the nucellus. This is the tissue where meiosis occurred. Inside that region is the developing female gametophyte. They are attached by vascular tissue to provide nutrients as it develops. Focus on spatial relationships, where each structure is positioned relative to the ovary wall and locule. This entire structure is enclosed and protected inside the sporophyte. Now create a label drawing in your lab notebook. Draw the ovary wall and the locule for context. Then draw one ovule in detail. Label the ovary wall, locule, ovule, and megasporangium. This is not just about labeling, it's about understanding structural organization. Pause the video while you complete your drawing. To summarize, the female gametophyte is highly reduced. It develops entirely inside the ovary. Of the 4 meiotic products, only 1 survives. The sporophyte protects and nourishes it. This reflects efficiency and protection, 2 hallmarks of angiosperm success. Now we move to the male side of reproduction. You'll examine a cross-section of the Lilium anther. This is where pollen develops, the male gametophyte. Unlike ovules, pollen is produced in massive quantities. As you observe, think about how this contrasts with ovule development. Inside the anther are microsporocytes. Each undergoes meiosis to form 4 haploid microspores, the tetrad. Unlike ovule development, all 4 usually survive. These developing spores are surrounded by nurse cells, often called the tapetum, which provides nutrients. Eventually, the anther opens by dehiscence, releasing pollen. This is a quantity-based strategy. Before observing, answer this question. Why do angiosperms produce such large quantities of pollen if only a small fraction fertilizes an ovule? Think about probability. Pollen must travel. It may never reach a compatible stigma. Producing large quantities increases the chance of successful fertilization. Write your prediction before continuing. Now we'll observe the anther cross-section. At scanning power in the left image, locate the anther wall and identify the pollen sacs. Inside each sac are regions of microspore formation. In the right image, look for tetrads, groups of 4 haploid microspores formed after meiosis. In the left image, the microspores are still early in development. Identify the nurse cells, or tapetum, surrounding developing spores. These provide nutrients. Also look for evidence of dehiscence, splitting of the anther wall for pollen release. In the lily flower video, the anthers were in the process of turning inside out to reveal their pollen. This is what it looks like on a microscopic level. Compare this organization to what you saw in ovule development. Notice the difference in quantity and arrangement. Now create a label drawing of the anther cross-section. Draw one pollen sac in detail, label the microspores or tetrad and nurse cells. Show the arrangement of cells within the sac. Again, focus on structural relationships. Pause the video while you complete your drawing. Now let's connect what you just observed under the microscope for both ovule and pollen development. First, compare location and function. Ovules develop inside the ovary, protected and enclosed. Pollen develops inside the anther, eventually exposed and released. How does that positioning support their role in fertilization? Second, both megaspore sites and microspore sites undergo meiosis, but the outcomes are not the same. In ovule development, 3 of the 4 haploid cells typically degenerate. In pollen development, all 4 microspores usually survive. Why would the plant treat these differently? Finally, think about reproductive investment. Plants produce very few ovules compared to the massive quantities of pollen. What does that tell you about male versus female reproductive strategy in angiosperms? This is where structure, development, and evolutionary strategy all come together. To summarize, pollen development contrasts sharply with ovule development. All 4 meiotic products survive. Large quantities are produced. Nurse cells support development, and dehiscence releases pollen into the environment. This reflects a probability-based strategy.